Good afternoon. Thank you for being here. I'm Philippe Chambon, the CEO of EG427. And let's get rolling. So we are a clinical stage gene therapy company. We're based in Paris. The technology is out of the series of the university there. Started in 2019. We raised a Series A and a Series B that we closed in February from under our partner and BPI as the lead for that series. For 20 people, and our first product, EG110A, is in a phase 1b/2a clinical trial in the US, and I spend a lot of time talking about this today. So what are we doing? We are not a rare disease therapy company. Our goal is really to treat chronic prevalent disease of the nervous system, and we believe we can do that effectively because of the unique characteristic of non-replicative herpes vectors. Our first product, EG110A, is a product that silences the activity of type C, sensory neuron, and its first application is around a series of pathologies of the bladder. Behind this, we have a second product, EG132A, which converts glutamate into GABA and hence shifts the balances from excitatory to inhibitory at the level of some neuron with application, of course, across spasticity, neuropathic pain, potentially focal epilepsy. And beyond that, we're building a broad pipeline, including on the central nervous system side of things. This is all possible because of the team. I'm not going to mention everybody on this picture. As far as I'm concerned, I spent most of my business life in the US being on the venture capital side of things there between 1995 and 2017. Then I came back to France, and I'm a young CEO because this is my first CEO job. The second person here, Cornelia Haag-Molkenteller, is critical to our success. She's our Chief Medical Officer. She's based in Irvine, California, and she's the person that developed Botox in exactly the indication that we're going after with EG110A. So she's been there, done that. What makes non-replicative herpes vector so interesting as tool for the treatment of chronic pathologies, and in particular in the nervous system? First, it's large vector. We can put up to 26 kb of genetic material in those vectors. Second, and that's linked to the particular tropism of herpes vector, they are uniquely suited to retrograde themselves into neuronal tissue. So if you inject them in a specific location, they will find nerve ending and retrograde themselves into the nucleus, stay there as an episome. It's an episome that never integrates, by the way. And if you're inserted in the right section of this episome, you should be able to get really long-term expression of your transgene. What has been shown also with those type of vectors is you can redose them because they don't generate a very significant immune profile. So you have this redosing ability. So the safety profile is rather unique in the gene therapy field. And they're also low cost from a manufacturing perspective. We estimate that once we're at scale with our first product, will be below $1,000 per treatment dose. With that, we're building a broad platform that's really a plug-and-play type of stuff where we have a series of vectors that can accommodate transgenes of different size. We do routinely transgenes that have more than 6 kb of genetic material in them, but we can go up to 26 if need be. And to give you a sense, I'm not going to show any of the data today in the interest of time, but this shows you the type of manipulation we can do with the transgene, but to put it for maybe making it more real, we've already done vector. Where we have 3 different transgenes expressed in the long-term expression region of the vector. We can also do vectors that have one transgene of the short-term expression and another one of the long-term expression, which enable us to combine, for example, CRISPR-Cas9 type of strategy where we delete an abnormal gene and then use the long-term expression section to replace that gene function. Those are the type of things that we can do, and that we have, in fact, vector in development to do those things. At this stage, the pipeline is most advanced with EG110A, which is in this phase 1b/2a clinical trial for neurogenic detrusor overactivity. We'll follow that with development in overactive bladder and interstitial cystitis. And as I mentioned earlier, we have a second product behind that will help us increase the concentration of GABA in specific location. So EG110A inhibits directly the activity of type C sensory neuron in the bladder. So basically what we do here, we inject our product in the bladder wall, exactly the same way as Botox is done. But here our vector will find nerves ending, retrograde itself into the DRG, and there express a protein that inhibits the vesiculation process of those type C sensory neuron, hence block their ability to communicate with the motor neuron. But with doing that, we preserve the function of the motor neuron, which is very different than Botox that acts at the local level, at the interface between the motor neuron and the muscle. And with this mechanism, we believe, and that's what we've shown through our preclinical study, we'll be able to avoid those chaotic involuntary contraction that leads to urinary incontinence, residual volume of urine in the bladder, urinary infection, and for example, in NDO patients, this is the second cause of death from infection in that patient population. As you can see, those are reasonably large patient population. NDO is almost 2 million patients across all its indication, and refractory overactive bladder, which is really the severe side of overactive bladder, is also almost 2 million patients. So the target product profile for our first product is the inhibition of those type C sensory neuron. We've shown that we are doing that in our preclinical work. Long-lasting efficacy, we expect at least 3 years of efficacy from one course of treatment. The theory of herpes vector, herpes virus infection is that it should last a very long time, but only clinical trial will enable us to clarify how long it truly lasts. We believe that will be as efficient or as efficacious as Botox. We won't have the impairment of the voiding function or increase in post-void residual volume, which limits, in fact, the application of the usage of Botox in indications such as MS or Parkinson patient, because these patients obviously have problems self-catheterizing. We had a very clean tox profile in our R&D submission, and we expect the safety profile in human to reflect that. We're doing the first clinical trial in NDO patient with spinal cord injury, and we're doing that in that patient population because their disease is set, unfortunately, and hence they have very low placebo effect. So it's a dose escalation study in patients, and it's ongoing at this stage. This study is done across 4 different sites. It's an all-comer type of study with patients, both male and female, between the age of 18 and 75, open label, 3 dose, low, medium, and high, 4 patients in the first, 6 and 6. We have 4 sites in the US, LA, Houston, Ann Arbor, and Philadelphia. And from a urologist perspective, it's really no different than injecting Botox in the bladder of the patients. It's the same equipment, exactly the same procedure as is currently done with Botox in that indication. The safety and efficacy endpoint of this study are at 3 months, and it is really based on patient-reported outcome in terms of episode of incontinence, as well as a cystometry measure of bladder pressure and a series of bladder function done also at this 3-month endpoint. So with that, we should have reasonably quickly a good understanding of the safety profile of our drug, as well as potentially some hints of efficacy. And with this, so the first cohort is enrolling as we speak. We've announced the first patient enrollment in February, and so far so good. We're continuing. And we'll have the second cohort during the summer, the third cohort in the fall, early winter, we'll have a series of pre-planned analysis, and we'll hope then to have early discussion with the FDA based on the result that we see. Like any other gene therapy trial, all patients enter into a 5-year extension study to follow them for the long term, and it will also give us a better handle on the duration of efficacy if we see any in this first study. So with this study, what we expect to see is restoration of continence, a strong safety profile, and this for numerous years. From a market perspective, the current market is really dominated by Botox and neurostimulation in OAB. And as you can see, given the limitation of current therapy, there's a vast number of patients that are currently not being captured by current medication. So with this, we're continuing to build a strong pipeline. We've done a lot of work on the CNS side of things, where we have some very nice finding in terms of location of our vector in different brain regions. For example, we get very efficiently retrograde transfer to the layer 5 of the cortex, also to the substantia nigra. So we're building vector to target neuroinflammation, broadly speaking, with that type of tool. With EG1328, the one that alters the balance between glutamate and GABA, we are looking at focal epilepsy, especially given the very exciting result that another company in the US had with cell therapy in a clinical trial where they apply their therapy at the local level of the epileptic lesion, and show that this increase in GABA delivered through their cell line worked very effectively. So here we have a different way of getting to the same issue. And we are expanding the underlying platform, as I mentioned, by putting multiple genes, playing with the length of expression and the like. All of our work is currently done at the research level in our research lab in Paris, and as well as through extensive collaboration around animal model, in particular, across Europe and the US. So in summary, we believe that non-replicative herpes vector have very unique feature that makes them quite different from AAV type of vector in terms of their ability to carry large DNA sequences, the safety profile that they have, the very local expression and then therapeutic benefit that they can induce. So there's a whole range of manipulation that can be done with them. Our first product target a large market opportunity across a series of indications, so it's clearly a several billion-dollar product if everything works. And with that, we have multiple other applications that we're investigating in the pain field or in the central nervous system side of things. Thank you very much for your time.
Thank you.